Welcome to today's class. Today we are looking at bacteriology, and in this bacteriology our focus is on gram-negative rods. And in this gram-negative rods we are looking at the family Enterobacteriaceae. This is the course contents. We look at the following contents on the topic of Enterobacteriaceae. However, we will first of all look at the general properties of Enterobacteriaceae before we now take these genus one after the other. Then we also look at gram-negative rods that are non-fermenters, which include Vibrio, Campylobacter, and Helicobacter. Thank you. So the family Enterobacteriaceae, they are generally lactose fermenters. We have also non-lactose fermenters. The isolating media is Maconche. They are one of the largest family of bacteria. Mostly they are pathogens, and their habitat includes human guts and environment. They are usually found in the intestine of humans and animals. They have over 100 genera and up to 1,300 species. They are straight rods. Some of them are motile, while some are non-motile. Most of the species grow well at 37 degrees, while some have an optimum growth temperature of 25 to 30 degrees centigrade. Now, their general importance includes the fact that some of them are harmless commensals, and we have some of them that are significant pathogens. And those pathogenic among them include Salmonella, Escherichia coli, Klebsiella, Shigella, and so on. So these pathogens will cause a wide range of infections, including gastroenteritis, UTI, pneumonia, septicemia, and meningitis, to mention but a few. Looking at their general characteristics as a family, they all have a general unique characteristics that we can say this is what makes them to belong to the family. That includes the fact that their cell wall are made up of lipopolysaccharide, and this is what is responsible for their staining characteristics, and that's why we classify them as gram-negative bacteria. They are all gram-negative bacteria in terms of gram reaction, and also they are facultative anaerobe, which means they grow with or without the presence of oxygen, and they can all ferment carbohydrate to produce lactic acid, and this allows them to adapt to anaerobic environment during fermentation. And all these all the family of these bacteria are all catalyst positive. Catalyst enzyme is present in their cell, which allow them to break down hydrogen peroxide into water and oxygen. This is a useful test to differentiate the family of Enterobacteriaceae from other gram-negative bacteria. This test can be carried out in a laboratory, and it's an easy test by dropping the colony of the bacteria in hydrogen peroxide. It gives off bubbles which indicate that they are positive. Now, they are all negative for oxidase tests. So oxidase is not present in their cell, and this is another useful test in distinguishing them from other gram-negative bacteria. These are one of the tests that can be carried out immediately you suspect that isolate is Enterobacteriaceae. So it can help us to differentiate them from other organisms, because the cultivating media also facilitate the growth of other bacteria, so it's not easy. And some other bacteria are also lactose fermenters. So the way to differentiate them fast is by carrying out catalyst tests and oxidase test apart from the staining characteristics. Now, additional characteristics of Enterobacteriaceae include the fact that they are motile. Most of these bacteria are motile. They possess flagella that enable them to move about in the environment. For Enterobacteriaceae, they have major types of flagella, which are three major types. We have the pertuticious flagellation, which is common among some Enterobacter species, which is described as a kind of covering. The flagella is all over the cell of the bacteria and it's distributed evenly. It allows them to move in a smooth and tumbling motion. We also have another type of flagellation which is called pertuticious flagellation, which means that the flagella are arranged at one or both ends of the bacterial cell. So it also allows them to move in a more directed manner. These are common among Salmonella species. And Salmonella are also one of the important members of this family. The other type of flagella that is also common is monotricious flagellation, which is characterized by one single flagella that is attached to one end, either the posterior or the anterior end of the cell. And this is also common among Proteus mirabilis. Characteristics of Enterobacteriaceae is their fermentation ability. Now, Enterobacteriaceae ferment a wide variety of carbohydrates, which includes sucrose, lactose, and glucose. Now, this fermentation ability is used in differentiating the members of this family, especially on their growth colonies on Maconche agar. Maconche agar is a differential media that allows colonies of bacteria to be detected and differentiated from one another based on their fermentation abilities. Those that can ferment carbohydrate has a pink coloring as shown in the diagram, while those that do not ferment has a colorless coloring. Now, those that ferment, we call them lactose fermenters, while those that do not, we call them non-lactose fermenters. So this fermentation ability is one of the broad way in classifying them into two different parts during identification. Example of the colony that we're talking about and those organisms in these categories. Now, those active fermenters include Cetobacter, Escherichia, Enterobacter, and Klebsiella. Now, there's an abbreviation to identify them which is called SIC, and this is used in differentiating them from those at non-lactose fermenter. You can see the pink coloration here. Now, and this is numbers of the non-lactose fermenter. You see the colony there, they are lactose negative. We have Shigella, Yersinia, Proteus, and Salmonella, which are non-lactose fermenter members of Enterobacteriaceae. Now, you need to know this that these listed ones are just the medically important Enterobacteriaceae. Like I said, you have about up to 1,300 species, some of which have not even has any medical significance. It also has the ability to reduce nitrate. Is one of the functions, characteristics of Enterobacteriaceae, which has the nitrate reduction properties, which distinguish them from other family as well. And the antibiotic resistance, Enterobacteriaceae are becoming one of the increasing resistant bacteria that are known, with members such as Klebsiella, pneumoniae, Proteus, and E. coli known prominently for their resistance, including Enterobacter. They are known to cause a lot of public health concern, especially in the hospital, responsible for nosocomial infection and also community-acquired infection. So one of the means of antibiotic resistance in Enterobacteriaceae include acquisition of mobile genetic elements, which include plasmid, transposon, and integrons, which allows them to transfer this resistant mechanism via horizontal gene transfer to other related family or another family of bacteria such as Pseudomonas aeruginosa, and it's one of the problems that is globally um, known, and we are currently battling with that as humans. They also possess antigens. These antigens are properties that contribute to their adherence and invasion of immune system and immune responses. They possess antigens such as K, H, and O, and these are one of the properties. So their genome content also possess virulent factors such as toxins, adhesins, and invasins. These are examples of um, properties which allow them to be virulent, to make them to cause disease, make them to uh, increase their infectious abilities, leading to situations such as diarrhea and all that. So their properties are general, cut across the organisms. Now, these are the tutorial questions that I have. We can help you in answering the questions regarding general properties of Enterobacteriaceae.